Hey, what's up, biochemistry classmates? Today we're going to be talking about the objective set Cholesterol Metabolism 1. There's a part 2 that I will be posting tomorrow morning. Um, just a little update. So, for complex lipids 1 and 2, I actually made videos for those, but I was not happy with them. I did the reading 21.2 and 21.3, but I realized that those readings weren't quite enough to answer those objectives well. So I will be remaking those videos, and I'll be using the lectures that the lecture slides that he gave us, in addition to like chapter 10 in our textbook. So I will be remaking those. Those, however, will not be up soon because after these two videos, I will start working on a Unit 2 review. One of our classmates actually requested that because it's been so long since we learned it. So I actually think that would be a good idea to make a Unit 2 review. It'll be really condensed and short though, so definitely start studying for that on your own first. Um, oh, and today I have like a different format. I don't know if you could tell, but so initially I was using Photoshop, and I was aiming for like a Khan Academy type thing. So I kind of just wanted to try this OneNote out. I actually liked taking notes on this a little bit better. It was a little easier. Um, doesn't have the same color scheme, but I actually like this quite a bit as well. So, anyways, let's get started here. The first objective is know the structures of cholesterol and its hydrophilic slash hydrophobic moieties. Okay, so cholesterol has the word sterol in it, right? And so it's going to be a derivative of a sterol. Essentially, what a sterol is a four fused carbon ring. So here you see 1, 2, 3, 4. Three of them, right? 1, 2, 3, are going to be 6 carbon rings, and then the last one, the fourth one, is going to be a 5 carbon ring. Okay. Okay, so these fused rings, right? They're going to give sterol a planar, so flat, and rigid structure. Okay. Um, so cholesterol is just a derivative of a sterol, and this is what it looks like. Know how to draw it or recognize it. Um, the OH, that's the polar moiety, and then everything else is the hydrophilic moiety. All right, objective B. Know the three general functions of cholesterol. Okay, so this first function we kind of went over in 445. It was actually a test question and I got it wrong, and so I will remember this for the rest of my life. It's going to be the last thing I think of on my deathbed right before I die. Basically, cholesterol will Basically, cholesterol will associate with a sphingolipid, and these sphingolipid cholesterol combinations are gonna make your cellular membranes more rigid. Or if you remember this notation, L naught stands for ordered. So yeah, this is gonna give it a rigid structure. Um, the second function is that it's a precursor to many steroid hormones. We'll talk about this a little bit more, but the important ones are testosterone, estradiol, cortisol, and aldosterone. We'll talk about those later. And the last one is it's a precursor to bile acids. So bile acids are made in your liver, but they're stored in your gallbladder. And when you eat something really fatty or anything that has fat in it, your gallbladder is going to excrete bile acid into it. Another word is called taurocolic acid. And essentially, this guy is going to act like a detergent and form these little micelles with the fat that you just ingested, which is an important step in sort of absorbing the fat into your body. We talked about this in Unit 2. So once I make the Unit 2 review, you could check that video out and kind of tie these concepts together. All right, objective C. Know the 4 stages and reactions for cholesterol biosynthesis. So there are obviously, right, 4 stages to cholesterol synthesis. There are actually a lot more steps though, because there are multiple reactions for each of these steps. So we'll do the 4 stages first, and then we'll get a little bit deeper and explore the reactions. The first step is going to be a formation of mevalonate from acetate. I don't know if I'm saying that right, so we'll call it mevalonate. And then you're going to use your mevalonate, and you're going to create activated isoprene units, right? And then from there, you're actually going to use 6 of these guys. So isoprene units are 5 carbons long. And so we're going to use 6 of them, and we're going to produce what's called squalene or squalling. So that gives us a 30 carbon molecule. And cholesterol is 27 carbons. So we're going to have to, first of all, make it cyclic, right? Remember, there are 4 rings. And then there's some like cleavage and like oxidation that happens after that. So here is a picture from the textbook that shows you each 4 steps, right? Acetate to mevalonate, and that goes into activated isoprene. And then these activated isoprene units create squalene, and then they cyclicalize. I don't know if that's a word, but they cyclize and they make your cholesterol. So let's explore each step in further detail. Okay, so synthesis of mevalonate from acetate or acetyl-CoA. So we're going to start off with 2 acetyl-CoAs, right? And we've seen these two, actually we've seen all of these enzymes before in, I believe it was ketone synthesis. Don't take my word for that, but I think this is where we've seen those. So we're going to use 2 acetyl-CoAs, right? We're going to use acetyl-CoA acetyltransferase. We're going to get a free CoA group, and we're going to make this acetoacetyl-CoA. All right. So we've seen this guy before in ketone synthesis. Then we're actually going to add another acetyl-CoA group. You see this? And we're going to use HMG, which stands for beta-hydroxy beta-methylglutaryl-CoA. So HMG-CoA synthase. This is going to attach another acetyl group to our growing chain, right? And then after that, we use HMG-CoA reductase, and we're just going to basically reduce this guy. So where does the reduction happen? Do you see it? Do you see it? Do you see it? So the first reduction is the fact that we lose this sulfur bond, so we get an H there. And the second is this ketone turns into a hydroxy group. So you'll notice here that we need actually 2 forms of redox currency, so 2 NADPH, and it'll yield 2 NADP+. We'll also get a free CoA from this, all right? And then we made our mevalonate. This is the commitment step in, I think that was one of the objectives, right? Oh, I guess not. Well, this is a commitment step in cholesterol synthesis. So this is where regulation occurs. So know this step, this reduction step right here that requires 2 NADPH. Here's a little acronym. Remember TSA. So the second 2 are HMG-CoA, and the first is acetyl-CoA acetyltransferase. So TSA, like when you get checked out at the airport, is for transferase, synthase, and reductase. And after that, all you have to know are the 2 names or products and intermediates that are used to name those enzymes. Okay, step 2, conversion of mevalonate to 2 active isoprene units. So you start with your mevalonate, right? You're going to use an ATP, and you're going to use the enzyme mevalonate 5-phosphotransferase. All this is going to do is add 1 phosphate group to your hydroxy group out here, right? Then you're going to do it again, this time with phosphomevalonate kinase. So this first one was a phosphotransferase. This one's a kinase. They do similar things. And you're going to get a second phosphate out here. So this mevalonate became a 5-phosphomevalonate. That makes sense, right? Your fifth carbon's right here. It's not letting me write. Your fifth carbon's right there. Here you use another ATP for another phosphate group. Now you have a pyrophosphate, right? So 5-pyrophosphomevalonate. Then you're going to use or pyrophosphomevalonate decarboxylase, all right? And essentially you use another ATP and out pops a CO2 group, right? And a inorganic phosphate group. So what happens here, this is an intermediate right here. So really this right here, oh, where did it go? Oh, so from this arrow to this arrow, that's actually that's one step. This is just a intermediate, or not an intermediate. What are these called? The things that don't last. So yeah, so this is what I did here. I just basically condensed all of it, right? In goes ATP, out comes ADP and CO2. So all you're going to do is cleave that guy, right? Or cleave that guy. And then ATP, I guess, is used as energy. And you're left with this, a delta 3 isopentenyl pyrophosphate. Now this will be in equilibrium with dimethyl allyl pyrophosphate, and your IPP isomerase is going to be responsible for interconverting between these two. So actually both of these are activated, and both of them are going to be used to make squalling. So let's move on. Let's see how we make it. So condensation of 6 activated isoprene units to form squalling. All right, so we start off with each of our activated isoprene units, right? So remember up here we made 2 isopentenyl and then also dimethyl allyl. And they're basically just going to combine. You're going to lose a pyrophosphate group, and this is called head-to-tail condensation. So it's exactly what it sounds like. The heads and the tails will combine, right? You're going to use a prenyl transferase for this, and you're going to get geranyl pyrophosphate. Essentially, you're going to do the same reaction, right, with another IPP, and you're going to get a 15 carbon long farnesyl pyrophosphate. So geranyl and farnesyl, these names aren't scientific. Geranyl is the oil that's found in roses, I think, that gives it its nice smell. So that's where we first found this intermediate, was in a rose oil, and so that's why it's named geranyl. And farnesyl is named for a similar reason in a different plant. But anyways, so we're gonna use 2 farnesyl pyrophosphates from there, and we're gonna use some redox currency and squalling synthase, and we're just gonna combine these two. So we're gonna combine this group, and we're gonna combine this group, and we lose 2 pyrophosphates. 1, 2, or I'm sorry, 1, 2, because pyrophosphate has 2. You're going to combine them, and you're going to get your squalling. Now, conversion of squalling to a 4-ring steroid. So this is your squalling. You can actually rotate about these single bonds, and you get something that looks a little bit more like this. So it looks like a steroid group that's like kind of has gaps in it, right? So you're going to use a squalling monooxygenase with redox currency and O2. And you're basically just going to oxidize this thing and make squalling 2,3 epoxide. I don't know if you guys remember some of your OChem. From here, this guy's going to branch off depending on what species you are. So fungi use it to make this, right? And then plants make it to use this. But for us, we use cyclase, right? And we're going to create this group right here. And this group is going to turn into lanasterol. So cyclase, and then we make lanasterol, and it's going to cyclize in this step right here. This right here is already a steroid, right? Because it has a sterol group in it. But then we're going to take extra measures, right, to oxidize some groups, reduce some other groups, and we're going to get cholesterol. Doesn't clarify how we do it, it just says multi-step. So just notice here we use O2, so it's an aerobic process. All right, objective D, we're almost there, guys. Know the rate-limiting step for biosynthesis of pregnenolone and structure/function of testosterone, estradiol, cortisol, and aldosterone. So we've already talked about that a little bit earlier. These guys are going to be our steroid hormones, right? Pregnenolone is just a precursor to all of these steroid hormones. Okay, so in the textbook, it provides us the pathway to from cholesterol, to pregnenolone. Essentially, you're just going to use a monooxygenase twice, right, with a cytochrome P45. We talked about this in one of our previous videos. I'm not going to get into it here, but this is like important in a lot of oxidation reduction reactions. This cytochrome group right here. So we do it twice. Use NADPH twice, right? It's also aerobic, right? O2, and we're going to get this 20,22 dihydroxycholesterol. From there, we're going to do another oxidation reduction, and we get our pregnenolone. So just know monooxygenase twice, and then desmolase in the second step. Know the structures, know the names, know your reactants, right? Know your products. This isocaproaldehyde, right? That's a product, right? That's released in the second step. And the objectives ask us to know the rate-limiting step. The textbook did not clarify or say anything about which one of these were rate-limiting. If I had to guess, however, it would be this first step, I think. Mostly because this is a flavor protein, and the book talked about how it was like a pretty big complex, so I don't know. My guess is this step, but it didn't clarify, so we'll have to wait till lecture for that one. Oh, by the way, guys, so make sure you check the description for all of these videos because I put updates in there, corrections in there. So, for example, which is rate limiting, I will clarify once I find out from like lecture or whatever it may be in the description. So make sure you check the description every time you watch my videos. Pregnenolone is a precursor to all the steroid hormones. We said that earlier, right? So testosterone and estradiol. So testosterone is a type of androgen and estradiol is a type of estrogen, or I think that's how it is. Maybe they're just like synonymous, but they are responsible for secondary sexual characteristics. So secondary meaning anything that doesn't have to do with your reproduction system. So I guess like being able to grow a mustache, or like having, I don't know, like chiseled jaws or like a deep voice, stuff like that. Okay, cortisol. This is going to affect your protein and your carbohydrate metabolism. Also, so that's the first function, right? Second function is it suppresses your immune system. And so what happens when your immune system's suppressed? You're also going to suppress inflammation and things like allergic responses. Okay, aldosterone. This is going to regulate your reabsorption of sodium, chlorine, and bicarb in your kidneys. The book said kidney, but we have two of those, so kidneys. Oh yeah, know the structures. This was early in the textbook somewhere, like page 300 something. We just have to know these four, right? Okay, objective E. Know the biosynthesis of 1,25 dihydroxycholecalciferol and the general mechanism by which it regulates blood calcium. This wasn't in our reading. I had to actually find it in the index, and we're going to refer to page 914 for this. So this 125 dihydroxycholecalciferol is a fancy name for a calcitriol, so we will call it calcitriol from now on. It's made from vitamin D, and vitamin D can be ingested through your diet, or it can be made by the sunlight, as a lot of you may know. So we have this 7-dehydroxycholesterol in our skin, and basically when this guy's exposed to UV light, it will become vitamin D, cholecalciferol. All right. So it didn't have a detailed pathway of how this is done. This is all I had in our textbook, so I would just know this probably. Once again, check the description. If in the lecture Dr. Brian gives us a more detailed pathway, of course, in the description, I will talk about that so that you guys aren't left behind. So essentially, you're just gonna hydroxylate, so add water, right, or add OH groups to your vitamin Ds, and you're gonna get this dihydroxy. So you're gonna do it twice, right? And you're gonna get calcitriol. So calcitriol affects your calcium levels in your blood in two ways. The first way is it works with the parathyroid hormone to regulate mobilization or deposition of calcium from your skeletal bones. So if your calcium levels are too low in your blood, then it's gonna activate mobilization, right, with your parathyroid hormone, so that you increase your calcium levels. If it's too low, then it's gonna deposit onto your bones and make your bone kind of stronger, I guess. The second way is that actually activates the synthesis of the calcium binding protein found in your intestines. This is important for uptake of calcium through your diet. So two ways calcitriol works to affect your calcium levels. Last objective, and I actually drew parentheses over here, rate limiting step for the biosynthesis of bile acids and how the enzyme is regulated, and also the structure of bile acids. So refer to these two pages. This was in our reading 842, 374. That was in the past, chapter 10, I think it was. So I looked everywhere in our textbook for this thing in parentheses, the biosynthesis of bile acids. Couldn't find anything. I'm really sorry. So if in lecture Dr. Brian shows us what the rate limiting step is and also the biosynthesis, then I will include that in the description. But for now, all we have is its function, right? Or we have a little bit. Okay, so bile acids are synthesized from cholesterol, pretty obvious. And they act as detergents to emulsify fats obtained from diet. We talked about this earlier, right? This is kind of like the first step in getting that fat into our system when we eat it. So if you guys remember from OChem, detergents have a nonpolar part and they have an ionic part. So the example given to us in the textbook is taurocloic acid. I don't think the book actually provides any other examples, so it says know the general structure of bile acids. Just know this guy probably would, and you'll be safe. But if not, then you have this sterol group with 3 hydroxy groups, and then a taurine group with a negative sulfur group out here. So how I would recognize this is this chain with a nitrogen and a negative SO3 out here and 3 hydroxy groups. All right, so that's pretty much all for cholesterol 1. I will be working on cholesterol 2. Hopefully it's out tomorrow morning. I was skimming and looking at what it looked like. It looked kind of confusing. It's one of those multiple steps type things. Please share this with our other classmates. So I actually think I like this way better, using OneNote. Let me know what you guys like better. So you guys aren't very active, so none of you subscribe or like my videos or comment. So Yeah, so comment, do you like Photoshop better or do you like OneNote better? I actually like this one better, I think. It's a little bit laggier, but it's easier to navigate. Um, and it's easier to like enter text in and put pictures in. Not that you guys care, but yeah, so just let me know what you prefer. Subscribe and so that you don't miss a video, and like my videos so that I know that these are useful. Yeah. So I'll see you guys in the next video.